So what do you mean by epistasis? It is the phenomenon resulting from the interactions between genes at different loci. So epistasis is the phenomenon resulting from the interactions between genes at different loci. So that's very important. At different loci, not in the same loci. Now, epistasis is the phenomenon resulting from the interactions, relations between genes at different loci. So to get a better understanding of this, now we are going to the definitions, further definitions, because there are two types, two categories of epistasis: dominant epistasis and recessive epistasis. From that we can understand this better. So what is dominant epistasis? When a dominant allele at a specific locus, when a dominant allele at a known specific locus, so let's say we know one specific locus, when dominant allele at a specific locus, right, the word alters is missing here. We have to include, that's a very compulsory word. When a dominant allele at a specific locus alters the expression of a specific gene at a different locus, it is referred to as dominant epistasis. So when a dominant allele at a specific locus alters the expression of a separate gene at a different locus, it is referred to as dominant epistasis. Actually, to understand that, let's examine or just discuss a little bit of an example that will help us to understand the definition. The example that's given in the resource book is about two fowl varieties. Now the color of these two fowl varieties are determined by two genes. Now, as usual, the normally color deciding gene, that is the gene, the dominant C allele, the dominant C allele is responsible for pigmentation, and the recessive C allele results in the absence of pigmentation. Now in epistasis, in this example, what is special is another gene at different locus. That gene comprising I, the gene I, is epistatic to this gene C. This gene I is epistatic to this gene C, and it suppresses the expression of the color. The dominant I allele that you can see actually prevents the production of pigments. But the recessive I allele, this shows as the recessive I allele, no? This one doesn't have the ability to prevent coloration. Now to understand this better, let's look at a few combinations of gametes. That is, we can see certain organisms. Capital C, capital I, simple I. Now to understand this, suppose we don't have this, right? We don't have this epistatic gene. Let's say we don't have it. What do you see? What can you say then? Capital C is there. Then what should you get? A colored fowl, right? That qualification is there. But what's surprising here is that this capital I, this gene, what does it do? It suppresses the expression of the capital C dominant allele. So when this fellow is there, it prevents coloration, even though the double dominant condition is there. So that is the understanding behind that. But on the other hand, the recessive allele of the epistatic gene that we are talking about, it doesn't have any effect in preventing coloration. Now this organism, it is a colored fowl. Even this organism having just one dominant C allele, it is still colored because the simple I, that recessive allele combination doesn't prevent coloration. Now this combination prevents coloration. Even though one dominant C allele is there along with one recessive C allele, the capital I epistatic gene's dominant allele, what does it do? It suppresses the expression of this capital C allele. Right? It prevents pigmentation. So refer page 109 in your resource book to get a better understanding of this. I can't take time to discuss the whole thing, showing crosses and everything, but I'll just give a rough outlook again. So what's the understanding behind this? Now in the F1 generation, what we have got is capital C, simple C, capital I, simple I. Now the surprising thing is that this capital I dominant epistatic gene, right, of that epistatic gene, this capital I dominant allele, what does it do? It suppresses the expression of the capital C allele. So this fellow is disturbing us. What is it doing? It's suppressing pigmentation. So this organism as a whole is white in color. Now actually, when, just for MCQs, just remember this. When two F1 organisms are crossed, what happens? When F1 organisms are crossed, what happens? We get the phenotypic ratio of white organisms to colored organisms in the ratio of 13:3. So you have to remember this for MCQs. This is a very probable question. When the F1 organisms are crossed with each other, we get phenotypic ratio, which is white is to colored in the ratio of 13:3. This is because of epistatis. Now you see, due to the effect of epistasis, phenotypic ratio actually deviates from the Mendel's principles for the normal dihybrid cross. Why? Because the F2 phenotypic ratio is getting altered from the usual 9:3:3:1 to 13:3. Now in the F2 generation, according to Mendel's laws, normal thing, right? Mendel's principles, we get 9:3:3:1. So this is actually deviation, right? Epistasis. When epistasis is there, that is transformed as 13:3. Now with the great understanding that we have, let's look at this definition again. When a dominant allele at a specific locus alters, remember that word alters, I forgot to write that again. Sorry for the inconvenience caused because it's very difficult to write on this phone. I'm not using a tab, so I'm using the phone, that's why it's not that clear. So when a dominant allele at a specific locus alters the expression of a separate gene, we saw that it was another gene, right? Separate gene. When a dominant allele at a specific locus alters the expression of a separate gene at a different locus, it is referred to as dominant epistasis. So the dominant allele, keyword dominant allele, at a specific locus alters the expression of a separate gene at a different locus. It is referred to as dominant epistasis. So remember the explanations that I gave before explaining the definition again, then you can really understand this better. So what is recessive epistasis? When a homozygous recessive genotype, when a homozygous recessive genotype, when a homozygous, remember those words, very important words, homozygous recessive genotype, recessive, homozygous, recessive and homozygous. So when a recessive homozygous genotype of a particular chromosomal locus. When you're considering a particular chromosomal locus, slow down, a particular chromosomal locus, when a homozygous recessive genotype of a particular chromosomal locus alters or masks the expression, it inhibits or alters the expression of a separate gene. How interesting this is! Of a separate gene at a different locus. It is referred to as recessive epistasis. Wonderful things occur in nature. So this is so surprising because when a homozygous recessive genotype, normally we think those recessive things are not powerful, right? We disregard them, right? Tall plants, short plants, right? We just disregard those alleles responsible because they are only expressed in recessive conditions. Simple R allele should be there. But the problem here is these things show some power for the first time, right? Because when a homozygous recessive genotype of a particular chromosomal locus alters or masks the expression of a particular of a separate gene at a different locus. Remember the word separate gene at a different locus. So a separate gene at a different locus, it is referred to as recessive epistasis. So once again, when a homozygous recessive genotype of a particular chromosomal locus alters the expression of a separate gene at a different locus, it is referred to as recessive epistasis. So I will show you some diagrams to understand this better. So remember this story before I explain. Once, right, let's say a scientist, he crossed two homozygous dominant purple flowers, right? Two homozygous purple, I'm sorry, he crossed homozygous dominant purple flowers with homozygous recessive white flowers, and he got 100% purple flowers. In which generation? In the first generation, obviously. Now surprisingly what he noticed was when he interbred those F1 plants, and when he got the F2 generation, actually he saw purple flowers and white flowers in the ratio that he never expected. He got the ratio of 9:7. Now actually what was he expecting? Now he was expecting Mendelian genetics to work. He expected 9:3:3:1, but surprisingly it changed into 9:7. Oh, so that was his observation. So how does this happen? Let's see the story behind this. Let's investigate the story. So what plants did he use? Capital A, capital A, capital B, capital B, simple A, simple A, simple B, simple B. This is the homozygous dominant purple flower, and this is the homozygous recessive white flower. And in the F1 generation, what did he get? 100% purple flowers. Purple flowers. But in the F2 generation, he got, when he interbred F1s, what did he get? He got a ratio of surprising 9:7, which is purple is to white. This is the ratio that he got. Actually the F1 plants are like this, right? Capital A, simple A, capital B, simple B. That's very simple genetics. Now we'll see. Let's see, as in the early example, the combinations. From the combinations, you can understand recessive epistasis easily. Capital A, capital A, capital B, capital B. What do you get? That's no problem. Purple plants. No problem at all. Capital A, capital A, capital B, simple B, purple plants. Now, recessive epistasis. So something recessive has to work, no? There's a key. That is, if a combination like this is seen, capital A, simple b, simple b. Now there's a problem. Now what is the problem? To actually understand this, I have to mention this. Purple flower color. The purple color in sweet pea flowers is actually governed by two dominant genes, that is A and B. Now both A and B alleles, this is the special fact. If you understand this, you can understand what is recessive epistasis. And this example, both A and B alleles this time are coding for compounds. What kind of compounds that are necessary for the expression of purple color? Right, these A and B alleles, the dominant A and B alleles, that is capital A and capital B alleles, are coding for compounds that are necessary for the expression of purple flower color. Now hence, what you can see is that the purple flower color will be there only when the both dominant alleles A and B are present. So then you can understand that this particular organism that has resulted is producing white flowers because you definitely need a capital B dominant allele. You have to have both, right? Now I'll show you some combinations of organisms which show white color. Then you can easily understand. So tell me, what's the problem with this first one? Right, we don't have a capital B dominant allele, so we can't produce purple flowers because if you listen to my previous expression, what is that? Purple color in sweet pea plants is governed by two dominant alleles, capital A and capital B. Both A and B alleles are coding for compounds that are necessary for the expression of purple flower color. Hence, purple color will be there only when both the dominant alleles, capital A and capital B, are present. So it's clear, isn't it? So here was the problem with the second one. We don't have a capital A dominant allele. Third one, what's the problem? We don't have a capital B dominant allele. Fourth one, what's the problem? We don't have either we don't have both, right? Capital A or capital B alleles. That's the problem. And not this one. Don't get confused with this. Capital A, simple A, capital B, simple B. This organism is purple colored, right? It's producing purple colored flowers. But because it has a capital A dominant allele, capital B dominant allele, because if they have these two, they can produce the proteins necessary, right? Now, did I record that? Capital A, simple A, capital B, simple B, because they have capital A and capital B dominant alleles. In this combination, they can produce the relevant proteins. These genes can help produce the relevant proteins and then give rise to a purple flower and give rise to purple flowers in a plant, right? So that's okay. So if you listen to this statement very carefully, what's the statement? Double recessive genotype at any one of the above locus is epistatic to either homozygous dominant or heterozygous condition of the other. What does that mean? Very simply, that is,